Buono.
Ok, informiamo la gente a casa che il seminario sta prendendo inizio. Grazie. Allora, dicevo, grazie di essere venuti, anche se siamo in pochi. Alessandria è una piazza difficile e speriamo che questo seminario possa interessarvi perché non è facile avere personaggi come Cico qua con noi. Io sono molto contento, vi dicevo, Cico è un professore dell'Università Federale di Rio de Janeiro io potrei definirti Cico un genetista molecolare, giusto?
Sì.
Che lavora però principalmente su specie che non sono umane, no? Per cui lavora sulla biodiversità e è anche professore qua a Lupo, all'Università del Piemonte Orientale, per cui lui ormai da quanti anni, Cico? Da
5 anni.
5 anni insegna nel corso di Food and Health, che è uno dei nostri corsi master a Vercelli, e insegna proprio ecologia, però che naturalmente immagino con un'impronta che sia sempre molto molecolare. Allora, il seminario di oggi sarà, vedete, un'affascinante storia sulla biodiversità, sugli studi molecolari, genomici, eccetera, di tutto quello che Cico ha fatto da 13 anni a questa parte. Ci parlerà del genoma degli uccelli, dei pappagalli, di queste tartarughe preistoriche che dicevi, e anche delle piante sacre sciamaniche che Cico sta sequenziando, che fanno parte della tradizione, diciamo, amerinda. E poi ci dirà lui meglio queste cose. Grazie Cico, lascio a te lo stage.
Ok, grazie mille Francesco. Buono, io non posso parlare italiano, quindi parlerò inglese. Grazie, it's a great honor to be here. Thanks a lot. And I will be showing, therefore, today, the work we have been doing in Brazil, in Rio de Janeiro, in my laboratory, about the genomics applied to the biodiversity, okay? So first I have to introduce myself. I am actually a biologist, and let me just move this like here. And I know about myself, but and then I have a PhD in bioinformatics. I studied in England. I made a postdoc in France, and I made another postdoc in Mexico. And I have been visiting also many other centers. But now I am professor of bioinformatics and philosophy of biology, theoretical biology, and also science and arts in Rio de Janeiro since 2011. I'll be showing you some of the work we have been doing in the Genomics and Biodiversity Laboratory, on which we have been studying the amazing biodiversity in Brazil using genome sequencing approach, right? So we find some organism of interest. We go there, we collect the sample of this organism, a piece of tissue or blood, and then we purify the DNA and we sequence the genome of those organisms to understand in each organism. I usually say that each organism is sort of superhero of something, right? So we have lots of superhero animals, and I'll show you a couple of those organisms, what we have been looking and discovering in their genomes, okay? We are always trying to understand their metabolism and the genomics. We look for duplications of gene families in these organisms because this normally show us why these organisms do the superhero powers that they have. And we always try to understand how a given genotype can explain a phenotype in the organism. And we make a sort of data mining with those data, okay? So we have been sequencing lots of birds and also fish and also plants and lizards and ants and whichever organism that there is someone interested in sequencing their genome, we do it. We do it together with the zoologists, with the botanists, and then we interpret and try to understand what the genomic data can tell us about those organisms. So we want to understand the molecular mechanisms related to the phenotypic representation of those organisms. We predict genes of interest. Okay, we see the genes that are under natural selection on those organisms. And for example, we have been studying, I'll be telling you about the genome of the parrot, the genome of a hummingbird, the genome of fishes, the genome of the golden mussel that I believe that people have already told you about it. I participate in the project as well. Then it's a pity that this works in genomics and biodiversity. Generally, they don't have too much interest of the people that give money because it's expensive to do it. It takes like 5 years at least to do a whole genome. And sometimes they don't have interest, okay? But sometimes they have, and then we can get money and sequence those organisms. So also it helps in the conservation biology. Okay, and some organisms, we can have a glimpse of some biomedical application. In that case, it's more interest for the financiers of the work. But we are biologists, and we want to understand actually the phylogenomics, the taxonomy, the systematics, to understand how those organisms evolve, where they come from, and what's the relationship between those organisms and other organisms in biology. Okay, so I'll be just telling you about a couple works we have done. This was a work we did about the origin of the turtles. The turtles, you may know, there are 2 groups of turtles. That is one that's named Cryptodira and the Pleurodira. There are 2 great groups of turtles. One has the neck that goes on shell like this, like it's called the hidden neck turtle. And the other is the side neck turtle, that the turtle like this one, it puts the head like this by side, right? And these are 2 great clades of turtles. This one is more biodiverse. We have 11 families and 208 species. And Pleurodira, only 3 families and 8 species. And there has been a controversy about the evolution of those groups. And then we sequence a couple mitochondrial genomes that we are specialized in sequencing the genomes of mitochondria. And we used also a new model for, so we used 147 species, data from 147 species of mitochondria. All the turtles that have the mitochondria sequence, we used like 147 species. So we align the sequence of all genes in all the mitochondria. And we made we use also the nuclear data, the data from the chromosomes, right? But the chromosomes, we have few data, so we have just 25 taxa. And then what's very interesting for turtles is that they fossilize well because they have this shell, right? And it's like a bone, it can be fossilized. So it's known lots of fossils of turtles. And we used fossilized birth and death model. This is a sort of new model. And we were capable to understand, to date precisely where these 2 groups of turtles, they split in nature. And it has been exactly in the starting of the Jurassic time, on which we have the continents splitting. It was exactly in the split of Laurasia and Gondwana, right? When the continents shifted, that those turtles also shifted. A group of them stay in Laurasia, and another group of them went to Gondwana. And this explains also why the Cryptodira, they are found in Americas and Africa, and Pleurodira are more found in Laurasia. So using like 60 different fossils that we knew the fossils, and we knew the time on which it was fossilized, we produced this new model using the molecular data, the fossil data, and computer work. So we found out that the turtles separated on that time in the beginning of Jurassic age. Okay. Another work that I want to tell you here is about a work we did also with bromelia. This is a plant work. We sequenced a very interesting model of bromeliaceae, that's named Vrisia, is the genus Vrisia, that there are two morphotypes. There is this morphotype that's a conical flower, this one. This one is pollinized by the hummingbird. The hummingbird put the beak there and gets the nectar, right? And this other one that's more open is pollinized by bats like Batman, right? The bats. Okay. And then there has been a great problem and difficulty for the researchers to understand what happened, because very similar species, they have the two morphotypes. Okay, the one pollinized by bats, that's the open flower, and the more closed conical flower, that's pollinized by hummingbirds. And then in this case, we did what we call the ddRNA-seq. That is an approach that we sequence a partial genome. But then, instead of sequencing partial genome, we sequence partial genome of almost 60 or 50 or 60 different species. So it was a work of Beatriz, that is the first author of the manuscript. She went and collected like 50 different species of Vrisia in the field in Brazil, in Rio de Janeiro mostly, and we sequenced a partial genome for each species. And then we compared those genomes, and what we found out? We found we want to understand what's happening in these morphotypes of flower, and we found out after doing this phylogenomics tree on which we use many genes, we found that the bat morphotype was ancestral. Okay, this here is the ancestral clade, and the Vrisia therefore was initially in the past pollinized by bats. And then at some point, when they came to a sort of different type of forest, more closed, it arose an entire clade, this orange one, that was pollinized by the colibri, the hummingbirds. So it has been a change in morphology in all this clade. And also we found out one special small clade here down, on which we have both morphotypes pollinized by bats and morphotypes pollinized by hummingbirds. And this was very interesting to understand the evolution of this organism. And they probably have 5.8 million years ago when this shift occurred and the plants started to be pollinized by hummingbirds. So all of this we discovered with genomic approach. Okay. So I'll be telling you in the next slides about the mitochondrial genome. So we have been working. What's the issue on mitochondrial genome? The mitochondrial genome, it's a very, I was about to say easy. It's not easy. Everything is difficult for us when we do molecular biology. But the mitochondrial genome is a circular genome because the mitochondria was a bacteria once, right? Was endosymbiotic, entered the eukaryotes in the origin of eukaryotes, but the mitochondria kept their genome, a small genome. That's like in animals in general is 16,000 of base pairs. This is not big, but it's not small. It's enough. They have the mitochondria, they have 37 genes in 13 protein coding genes, 22 tRNAs, and 2 ribosomal RNAs. So every organism that we get the DNA and we sequence, the first thing that we get is the mitochondrial genome because it's small, it's easy to assemble. We know the genes that are there, okay? And it's a genetic marker. We can study the evolution of organisms using mitochondrial genome. Sometimes the data provided by the mitochondria cannot be very trustworthy because it's very small and can be biased. But in general, it's a first approach when we want to understand the evolution of organisms. So we sequence the genome of the mitochondria, and we can do comparative genomics. And this was a paper written by Marcela. Probably you know Marcela. She has been here when she was doing her master's. I was the co-supervisor of her. And we have found a very interesting thing in this work. That's the mitochondria of the muscle, the golden muscle. And this is, for example, the Mytilus. And we see that in the genus Mytilus, all the order of genes in the mitochondria, this is the order of genes. It's circular, right? So we just defined that one is, the first is this, and then this is the order of genes. So in Mytilus, all the synteny, that's the order of genes in the genome, is conserved, right? So the genes is one after the other. This is the synteny conservation. It means that this clade is correctly placed and done. But then we sequence perna, that is a specific muscle. And we found out that the order of genes between perna and the similar one, the other organism, the same genus, Perna viridis, the order of genes between the two species from the same genus is completely different. You see here? The COX1, COX2, ND2, ND1, the order is different. But then, if we compare the perna to Brachydontis, it's exactly the same. And also here in the tree, it means that the genes, the sequence of the genes from perna are more similar to Brachydontis also. Okay? What does it mean? What does it mean? It means that probably perna is not a perna, it's Brachydontis perna. It has been probably a mistake in the classification of this organism. And this is not a perna, this is a Brachydontis. Or maybe this is not a Brachydontis, this is a perna, if you like. And this would be not a perna, but a musculista, something like this. Okay, so when we sequence the mitochondrial genomes, we can find mistakes in the taxonomy and systematics of species. And this only with the mitochondrial genome, we can have these ideas and this glimpse about. There was just one modification between Brachydontis and perna. That's the position of cytochrome oxidase 3. Okay, very well. Then we did the mitochondria of fishes. We did the mitochondria of nematodes. We did the mitochondria of psittacids, that the parrot and their cousins. Okay, and in those cases, the current systematics was okay. We just confirmed that the systematics, the classification of the organism was right, was correct, okay? And in many times, we get the 12 or 13 genes of the mitochondria, as I told you, and we concatenate all those genes and generate a big matrix and compare this matrix. This is called the super matrix approach. We concatenate genes and produce a big sequence with many gene sequences, and we compare and provide this tree, this phylogenomics tree. not phylogenetics anymore. And then after annotating like dozens of mitochondria from animals, we found out that what the researchers were naming the heavy strand and the light strand in mammals was mistaken. They were submitting the mitochondrial genome, and they were saying, oh, this is what we call the heavy strand. The heavy strand, in general, has more G's and T's, and it's named the heavy strand. And we found out that most of the mitochondrial genomes from vertebrates in the GenBank were mistaken. They were saying that the heavy strand was the light strand, and was the contrary. Okay, so we published also a manuscript about this and did a cartoon about this. So after sequencing the mitochondria, that's a first approach when we do genomics of animals, we started to study plants as well, because there were some interest in plants as well. This was the first plastid of an orchid sequenced, and we did it in our laboratory, but the plastid is 10 times bigger. The genome is like 160 kb, 106,000 of base pairs. And instead of having 13 protein coding genes, we have 79 genes, and we make the entire annotation, and we make comparative genomics, and also we did it. And also we have been sequencing as well the mitochondria of plants. The mitochondria of plants, in that case, has about 500,000 base pairs. Okay, the mitochondria of animals is small. The mitochondria of plants is big. So we sequence as well the mitochondria of a couple plants. And then I started small and I am growing. And now we did the entire genome of an organism, the entire chromosomal genome that was the chromosome of the parrot, the green parrot, and the Amazona aestiva parrot. Okay, we sequenced the entire genome, and we find out lots of specific genes of psittacids. We found that this organism is long-living, and then we found new genes of longevity. For longevity, we proposed new genes for longevity because the parrot is like this big, and it can live like 90 years old. There are some reports saying that this bird can live long. And then we made some comparison when we provide some genes, some new genes for longevity, okay? Also, the parrot, if it's a superhero, what does the parrot do? He speaks, right? It speaks. The parrot is the bird that can speak better and communicate probably better. So we found out also some genes that were very much selected by natural selection. Use this DNDS approach when we search for positions in the DNA that are modified more frequently. And we found that many of these genes that are positive selected in the parrot, they have to do with cognition. And we also found out that some of those genes that are positively selected in the parrot, they were also positively selected in humans, meaning that the evolution of cognition in parrots and in humans have, by accident, occurred in the same genes that are key genes for the evolution of cognition, right? Very well. Then we also approach another bird genome that's very interesting, this model. That's the bar-tailed godwit. The bar-tailed godwit is a bird that lives in Australia in the summer, okay? When it becomes winter in Australia, all birds that live in a beach, they live in a beach like this, a very beautiful beach. I went there to New Zealand, actually, and they live in a beach. And then when it gets like March and it starts to be like cold in Australia, they say, oh, I don't want to live in the cold. I will migrate. And then they go to China and they stay there for like a couple months, and then they migrate to Alaska, where they breed. And they go to Alaska when it's sunny there. They just want to live in the summer, okay? And in Alaska, there is a photo period of about 20 or more hours of daylight, because it's very north, okay? So there's daylight, and this daylight is a signal for them to reproduce. So they reproduce in Alaska, and after 6 to 8 weeks, the new chicks that had just hatched the eggs, the small chicks, they travel again to the same beach that the very same beach that they live, their parents live. They migrate again to Australia and New Zealand. So it's very amazing. That it's I think it's like 100,000 kilometers that these birds with 8 weeks of life, they travel together with the parents, their family, and they go back. This one they didn't go back because they just born, okay? But the fathers, their parents go back to the same beach. How do they know the way to follow? How do they know the beach they have to go? How do they know the time of departure? So we have been found in its genome that the circadian clock genes related with the day and night, the winter and summer, we have some genes like melatonin, right? The melatonin is this gene that we produce when it gets dark. And it's the gene to sleep, the protein that makes us sleepy. So they have lots of circadian clock genes, and they were positive selected in this species, right? And then I was a collaborator also to this paper that was a science cover in 2014. Okay, I am just here in the middle. It was a collaboration between the Americans and the Chinese, and we sequenced in that work 44 different bird genomes, complete genomes. I was involved in 5 of them. That was exactly the parrot, the bar-tailed godwit, and a couple other ones. And we provide a very complex analysis using different markers, only genes, only introns, only ultra-conserved elements. And using different genetic markers, we found different trees of birds, okay? So it was very difficult for us to understand which marker, which genes we must believe to understand the true tree for birds. So we use what we call the total evidence tree, on which we use exons from 8,251 different genes, okay? These were the genes that were single copy in each species, because there are genes that are 2 copies and 3 copies, but these 8,001, they were single copy, okay? And we use some introns from those genes, some what we call the ultra-conserved elements, that are elements in the genome that are very conserved, and nobody knows until nowadays what they are doing there. And we made a nucleotide data set of 41 million base pairs, representing 3.5% of an average avian genome. And with this set of genes, we built this tree that's even nowadays is probably the most relevant tree of birds that use all the birds. We separate first the doves and the dove-like. The gallo-answer that are the ancestor, the chicken and the other birds, and then the neo-aves, gallo-answer, neo-aves, and here in the neo-aves there is the doves and the passerelle, that's most of the birds there, and we also separate the land birds that we call core land birds to the core water birds. Okay, so the birds that went to water and the birds that went to earth, in general, they split very early on the genetic tree of birds. And this tree is still studied by many ornithologists, and they show it's very interesting to understand the evolution of birds based on that tree. That was very important in that study. Okay, well, then after working like 10 years with birds and other organisms, I come to work with plants, mainly the investigation about the genome of ayahuasca. Have you heard about ayahuasca? So ayahuasca is a beverage, sort of tea, that is used by the Amerindian tradition, the Amerindians, the Indians, to heal in ceremonies of healing. Okay, what means ayahuasca? In Quechua, that's the language of Peruvian Amerindians. Okay, aya means morto, defunto, espírito. Ayahuasca means liana, the vite, come diciamo in italiano? Vite. Quindi, so it means the liana of the spirits or the liana of deaths. And what's that? This is a beverage, sort of tea, that the color looks like the coffee, but it's more browny. And it's psychoactive. Psychoactive, it's psychedelic, if you like. It's a psychedelic substance that provokes an entheogen effect. And inside tells God, generate the God inside of you, okay? And the Indians, they drink it to see the spirits, and also in healing ceremonies where they heal your body, they heal your mind, and they heal your spirit, okay? And this is very true. And then we started with the genome of ayahuasca, the genome project of the entheogens. Besides ayahuasca, there are lots of other entheogens. And we did, at this time, ayahuasca and jurema, that's another entheogen, and we'll be doing other organisms. And how it works with the ayahuasca. There is a ritual that you spend in general the whole night in this ritual together with the indigenous people. But also in Brazil, there are some religions, institutionalized religions like the Santo Daime, União do Vegetal, Barquinha, and others, that they are syncretic religions that mixed Christianity with Indian thoughts and Indian beliefs. So at the same ceremony, a person can see Jesus Christ, and the other can see like the snake, the anaconda, and other entities. Okay, it's very common when someone takes psychedelics that they can see entities, and those entities, people in general, they report that they can sometimes talk with those entities, and the entities can give them some revelation. Okay, according to many reports from people. So in the ayahuasca ceremony, you take a doses about 50 to 100 milliliters, 1, 2, or 3 along the whole night. After 40 minutes, it starts to make the psychedelic effect. This is legal, right? In Brazil, this is legal. In Italy, it's not legal.
It's considered a drug.
It's considered a drug. But you know, even in this religion, the União do Vegetal, they enter in the Supreme Court of the United States, and after 5 years of judgment and all, the Supreme Court, the higher court of judges in the United States, they allowed them to use ayahuasca in a religion ceremony, because this is the freedom of belief. Okay, so it seems like in Italy and all the Europe now, it's not. There are some people from the União do Vegetal here in Italy and also from Santo Daime, but since 2 years, I think in the past, they are drinking water instead of drinking the whole sacred drink that is ayahuasca.
There has been a report on the newspaper, I think, less than 1 week ago, because there has been a claim by probably one of these religions, but our government, they refused. I don't know if the government of the Supreme Court, but
Yeah.
it is absolutely not legal.
Yeah, it's not legal. I gave
Even
this,
for religious purposes.
not even for religious purpose.
Yes,
Not even for religious purpose. Yeah, you know, well, this is something that we won't discuss here, but it's legal in Brazil, it's legal in the United States, and it's legal in other parts of the world. But here in Europe, it's not legal anymore. So the people drink the tea and they dance, they sing, they pray, and they make some concentration meditations, okay? And it's a ritual of purge, of cleaning and purge. What's a purge? It means that if you have something that's bad on your organism, you will put it out. So many people vomit, many people have diarrhea, many people cry, many people sweat a lot. And this is completely normal for this kind of ritual. And it's seen as a cleaning. You are cleaning yourself. Everything it's dirty, it's getting out of you, okay? This is the cosmo vision of the Amerindians. So regarding the genomics, we are talking about genomics, right? So the tea is made with 2 plants. That is Psychotria viridis, that is a chakrona, we call it chakrona. And chakrona is an arbust from the family of the coffee, Rubiaceae, the same family of coffee, okay? And then the Psychotria viridis present dimethyltryptamine, that is DMT, that is a psychedelic, non-psychedelic substance that you also have in your body. We humans, we produce endogenously DMT. We have the enzymes to produce DMT, right? But in that case, when we drink DMT, if someone drink DMT alone, the DMT is quickly broken by an enzyme that's named monoamino oxidase, MAO, okay? The monoamino oxidase breaks the DMT and transforms DMT that's psychedelic in tryptamine that's non-psychedelic. Then what happens with the ayahuasca? There is another plant is the liana, that's Banisteriopsis that presents, it's from the family Malpighiaceae, the family of acerola fruit, that presents an inhibitor of monoamino oxidase. So we have DMT. The DMT is broken by the enzyme monoamino oxidase. Okay, so one plant has DMT and another plant has an inhibitor of this enzyme that breaks DMT. So the DMT is active in the postsynaptic cleft for a long time. This is the scientific explanation for the psychedelic effect of ayahuasca, right? So these are some beta-carbolene named amines and amylines that inhibit this enzyme to break DMT, and so DMT is active for more time in the postsynaptic cleft. Right? So this is the, let's see, this is the serotonin, the molecule of serotonin, the molecule, and this is DMT. Look that they are very similar. The serotonin, we have here an indole, and then another carbon structure here. And here is the DMT. So DMT just don't have this OH here. And then this nitrogen here is methylated twice. That's what dimethyl is a tryptamine. So this is serotonin, that's the neurotransmitter of the happiness, if you like, right? And then the DMT works by binding the serotonin receptors, okay? And also, here we see the psilocybin. Also, it's very similar. Psilocybin is the molecule from the magic mushrooms. Okay, that's similar as well. Even the LSD is a sort of tryptamine as well, and then they bind the same receptor that they are the serotonin receptors. Okay. So the DMT is made by a decarboxylation of the amino acid tryptophan in your body and also in the plant. So we have a tryptophan decarboxylase that produces tryptamine, and this tryptamine is methylated once and twice. If it's hydroxylated, it becomes serotonin. If it's methylated twice, it becomes DMT. And DMT, there are some controversies, but it's expressed in your body, mostly in the kidney. We don't know why. And also there are some ideas that it's produced mainly in near-death experiences. When people that almost died and came back, they have some psychedelic as well, psychedelic views sometimes. And there are some ideas that they produce also DMT at that point. This is still a controversy, okay? So the DMT is now not only the DMT, but MDMA, the LSD, and the psilocybin is now becoming medicines. They are becoming medicines to treat some psychiatric disturbs, such as traumas, okay, for the war veterans in the United States that have trauma, they have been to war and they fight and they kill and they got killed and they lose friends. They have traumatic disorders, and it has been proven. It's still ongoing, this research, that the treatment with psychedelics can get them better, okay? Also for depression, there are many people, depression is one of the most important psychiatric disease nowadays, and there are many people that they cannot respond to medicines. They try one medicine and they keep being depressed. Then they change the medicine to another medicine and they are still depressed. So it's the depression resistant to treatment. So what's the last try that people make when these depressed persons, they keep being depressed? Oh, let's try the psychedelics. And then many times you try the psychedelic and the psychedelic can turn the person not depressed anymore. The person can get out of depression, okay? So there are lots of studies. This is not, this is very well studied, okay? There are lots of taboo about the psychedelics, we know, but science is proving that they work, okay? They need a different treatment because they change your consciousness, and you cannot be in this state of modified consciousness for long. You just made a trip, sort of psychedelic trip, and sometimes along this psychedelic trip, you can restructure your brain and your relationship with some key events in your psyche, and this restructuration can help you out of some psychiatric disorder, okay? So there are lots of works showing about the relevance of ayahuasca and DMT in psychiatric disorders, okay? Even for cancer, it has been tested, okay? And then we, of course, we don't work with the tea, but we work with the plants, okay, that produce the tea. And then we went to a plantation of chacruna, and we collected the plant. We did, in this case, the sequence, that's the Pacific Bioscience sequence, that the long reads, and we need to put the black structure around the black plastic to avoid plants to make photosynthesis for 24 hours. Then we can have a better DNA extraction of this plant. And then we collect the leaves and we put immediately in the nitrogen, liquid nitrogen. We freeze it. And in our case, we send to the United States, to the University of Arizona, I guess. And the guys there are the most specialists in the world to extract big DNA, big pieces of DNA. And then we sequenced, and we have a very high quality DNA and the sequence with high quality reads with very good quality, more than 99% quality in this reads. And we sequence, of course, first the chloroplast and the mitochondria of those plants. This work is published, okay? And then this first plant of ayahuasca, that's Psychotria viridis, that's the arbust, that is the leaf that we use, the leaf is not the liana. We sequence one lane of this plant, and we sequence 2 lanes, and then we found, oh, this genome is weird. What's happening? And then we start to make some studies, and we discover that this plant is an hexaploid plant. So we are expecting to spend some money with genome sequencing, and we have to spend 6 times more money because the genome is huge. So it's an hexaploid. They have like 6 copies of each chromosome. You know that humans are diploid, right? So we also predicted genes, and most genes are present in 6 copies. But also we have genes with 5 copies, 4, 3, 2, 7 copies. It's a very complex genome, and it was probably produced by both hybridization and autopolyploidization in this plant. So the haploid genome has 1.55 gigabase, 1.55 billion base pairs. It's like half of human genome. And the hexaploid genome has 6 times more. That would be 9 gigabase genome. Okay, it has 11 chromosomes, and this genome is duplicated, and some genes are lost. Some genes keep there, some genes are duplicated. And with a very new technique on bioinformatics, we were capable to separate the 6 copies of every chromosome. So we have 11 chromosomes, and we have 6 copies of each one. And we, with this new technique of sequencing in bioinformatics, we have most of them telomere to telomere. We have the whole chromosome sequenced. That this is very amazing. It was the work of a friend of mine, that's Alessandro Varani in São Paulo, that he did this bioinformatics approach that was very difficult, but he's a genius, so he could assemble the whole genome. And now, what we want to learn in this genome, we want to find mainly the methylases that will methylate the methylases that will produce the DMT. Okay, we are now working on this. We have the gene prediction, and we are searching for the methylase that will methylate the DMT, the tryptamine to produce the dimethyltryptamine. Okay, it's unknown which methylases do this, and we are working with all the methylases in the genome to find out which one is the right one. And then very briefly, we are sequencing the genome of the other plant, that is the liana, that's Banisteriopsis caapi. We have 2 different morphotypes. One that's the liana without nodes, and the liana with nodes. We went there to collect this plant as well. And this is the genome is more common. Okay, we have also the mitochondria genome, and it's a 1.3 or 1.1 gigabase genome, and it's 10 chromosomes. We have 2 varieties, Calpuri and Tucunacá, and we sequence telomere to telomere, the whole chromosomes of each. And in this case, what we want to, it's a diploid genome. Okay, so the first Malpighiaceae to be sequenced also in plant genomes. We have transcriptomes as well. Okay, so we have the full telomere, T to T, telomere to telomere genome assembled for all those species. Now we start the genome annotation, and we will have to find the enzymes that produce the beta carbolines, okay, the inhibitors of the monoamino oxidase. So there are also environmental issues. The chacrona is not, there's no least concern according to the IUCN. It's not a problem of conservation biology, but the liana, as it's more vulnerable, and we can also have some projects to help in the conservation of this species. Okay, yeah, this is our research group. We have been collecting those plants in the União do Vegetal. That's this religion. We went there. We took the plants. We took the tea with them as well. And it was a very interesting experience. Okay. Just to finish, I'll just tell you very briefly that besides work with the genomes, I work also with the origin of life. I've been here, I think 3 years ago, talking about the origin of life. And we have proposed 4 ages in the evolution of life: the prebiotic age, the origin of the ribosome. According to us and many other researchers, the ribosome was the first molecule in life. This is theoretical biology, okay? And then after the origin of the genetic code and the ribosome, that this was for us the origin of biology, we have another age that we call the age of progenotes, on which the genomes were assembled and also the biochemical pathways were assembled before getting to cells and organisms. Okay, so we have this idea of a chemical symbiosis between nucleic acids and proteins. the FUCA is the first RNA capable to bind together two amino acids. Is the catalytic site of the ribosome. This was probably the first living organism to exist. We have a manuscript also about the origin of the metabolism. And we tell the history of Earth, starting with the ribosome, then going to the progenotes that are nowadays extinct, then going to the viruses, and then going to the cells. This is a piece of our model. We have explained it step by step in a paper on which we challenge the current concept of viruses, because people think that viruses are obligatory cellular parasites, okay? And then we say in this manuscript and others that viruses, they don't need cells to reproduce. They just need a ribosome. They just need something to get their genetic information and produce proteins, okay? So we have a model to explain why viruses for us were originated before cells, okay? We have written a book, The Emergence of Biological Systems. And also we have in our laboratory a part that's for science outreach, that we make theater and music to teach science for the young people. Okay, so we have the Laboratory of Science, Arts, and Education, on which we do comic books, animations. I have a course that's a workshop of science, arts, and education, on which the students are invited to be inspired by scientific knowledge to produce drawings and charts and music. We have been doing some storytelling about the history of ribosome, mitochondria, chloroplast, and we go to schools to teach them in a ludic way, okay? So this is the theater part. This is me dressed as Aristotle, okay? So we go to the schools to present some theater. Here also another spectacle about the vaccine rebellion, and also this is the children that we are in the school in Rio de Janeiro, and we did a theater also about the dengue, and music. We have a band. And that's it. And just to finish, I've been here since 2019, teaching ecology to the students of the Masters in Health and Environment. And this course here also inspired me to develop an environmentalist course that I did in Brazil, where I speak about the IPCC, the International Panel of Climate Change, the ecological footprint, vegetarianism and veganism, reforestation, agroforest, and lots of themes linked to environmentalism, including shamanism, okay? So thanks a lot. It's a great pleasure to be here. Thanks, Francesco. Thank you all for watching. And I'm available for questions. Thank you. I have two questions. But I can read. No. Yes, stand up and write your question. The first question is, what protein generate this gene that produce the long life in the earth? Oh, there are lots of genes. And some genes, what we have, what was this analysis? This was, we compared the lots of birds with different times of living, and we found some genes that are specific from long-lived birds. Those genes, some of them are unknown. We don't know which protein. There are some sort of unknown genes until nowadays. And some other genes are genes from the metabolism, from the standard metabolism. There is a list. There are like 100 genes that we proposed as new genes related to longevity. So there are lots of them. I cannot tell you a specific one, but in the manuscript, if you like, you can get the manuscript, and there is the list of the genes that were probably linked to the longevity, not only in the parrot, but in other long-lived birds as well.
The second question is interesting that the DMT have an effect to reduce the cephalera. What is it?
Yeah, nobody knows.
Excuse me, to reduce what?
The cephalea, the headache. Headache. Yeah, the headache. We don't know. So there are, you are right, the DM, not only DMT, but other psychedelics also are used for strong headaches, that people have headaches and they don't know why. But what we know about the theory about psychedelics in general, and for DMT is the same, is that they promote neuronal plasticity, synaptic plasticity. I mean, what happens? You have in your brain a sort of synaptic network. If you remember your father, you just go from this neuron to activate this and this and this, and there is a sort of network or a graph that neurons that are activated when you think about, I don't know, a microphone. If you think about anything, you activate in general the same route of signaling in neurons. What's the theory for psychedelics? The theory for psychedelics is that they reboot, they restart this neural network. And you can think in your dad in using another route. So somehow you can reposition yourself and your relation to someone else or to some event in your past when you take psychedelics, and you can understand that from another point of view. So this is reconfiguration of synapses and synaptic routes. Also, the psychedelics, many of them, and DMT for sure, they are also neurogenic. They allow the creation of new neurons, the duplication, the replication of neurons. This is scientific, okay? There are many works that confirm this. And therefore, including in the case of the DMT, it has been proven in rats that some old rats that are already senescent rats can learn again. They say that the name is critical period. The critical period is the period on which you can learn something. After you or someone or any animal gets old, you lose your desire to learn new things. You just want to do the very same things you have ever done. This is called the critical period. You miss the critical period. But if you use psychedelics, you can reopen the critical period and you can be interested in novelties again, something new. So there are lots of different mechanisms that explain the effect of psychedelics. But then in the case of the headache, that's your question, you never know. But probably it's because of this, because of creating new neurons and also reconfiguring the synaptic network of your brain. So this in general is the explanation about the effects of psychedelics in psychiatric disorders, that they help people to recover. Okay, you're welcome.
Just to keep going the discussion on DMT, there is a question from the people online. How could exactly DMT work against depression? Can you add more, Chico, or?
Well, the mechanisms, it's still very enigmatic, the mechanisms. What people know is when, thank you. The mechanisms in general, they're explained for you guys in the internet. The mechanisms are explained by synaptogenesis and neurogenesis. So reconfigure the synapses, the network of synapses in your brain, and reconfigure and creating new neurons. But when you say about depression, there is a great group of Brazil that just work with DMT and depression. It's the group of Professor Draulio Barros de Araújo. It's a great group working on this in Brazil. What they do is giving DMT to the people, okay, and giving a placebo, and seeing that the people that took DMT, they recover from depression more than the people that take the placebo, right? We don't know exactly, the mechanisms, but we know with experiments in humans that the people that take DMT, they are recover like 50% or even 60% of the people that take DMT in this clinical trial. This is a clinical trial, okay? They get people with depression, long-term depression resistant to treatment, and they give them DMT, and they give them sometimes ayahuasca, okay? And they analyze the data. So they just know that the people recover, but the mechanisms, they are still enigmatic, okay? That's what I can tell.
Most of the research on, let's say, treating, curating brain disorders doesn't rely on a known mechanism.
Yeah, exactly.
And there is a lot of literature, I'm saying that for the people at home, on LSD, for example.
Of course. Yeah.
A lot of interest in this particular molecule. Okay, Chico, there is another question about the parrots, which is something general. I think, are there any studies about the ability to speak of parrots?
So what we discover in the genome, analyzing the genome, that there are many genes related to cognition. There are exactly genes related to neurological functioning, that they were more mutated in parrot. They have this natural selection, DNDS, operating in the parrot. So we know that those genes, that it's not exactly related to language, to speech, but related to cognition, related to brain functioning. So
when you say there is is there any bias when you count those genes related to cognition?
No,
do you carry out kind of a pathway analysis so you can map the number of genes into specific pathways, or how does it work?
no, it's a more analysis of mutations. We make multiple alignments, okay, of all genes of the parrot, and all genes we make multiple alignments, and then we see which genes they have this mutations that change the amino acid. This is the DNDS. That is the synonymous versus non-synonymous mutation. Okay, this shows that it has been a positive selection in a couple of genes, so and then we have a list.
Comparison between the parrots and other birds, or
yeah, in that case we compared long-lived birds with short-lived birds. And then we found...
You see that are more active.
Yeah, they are more mutated in birds that live longer.
Yes, active in the sense of...
Yeah.
That's very interesting. Okay. Avete altre domande? No. Posso?
Grazie a tutti.
Grazie mille.
It's a pleasure to be here.
Altre domande, vi ringraziamo per essere venuti. Chico sarà qua anche, speriamo, più spesso e più a lungo. Grazie ancora, arrivederci.
Ciao, grazie a tutti.
Ciao a casa, Grazie.
Arrivederci, grazie.
Grazie mille.